First, we're going to have Jinhan Yu. Jinhan is a graduate student in the lab of Professor Donna Blackmond at Scripps. So Jinhan got her bachelor's degree at Nankai University, and she performed research focusing on asymmetric catalysis, apropos. And following Jinhan, we'll have Neal Deng. Now, Neal is a postdoc in Professor Blackmond's lab. And Neal also started his education. He obtained his bachelor's at the Southern University of Science and Technology in China, where he also researched asymmetric organic catalysis. He then got his PhD in chemistry at the University of Connecticut in 2021 with Professor Gail Ung, studying this circularly polarized luminescence from chiral emissive complexes. So this really is quite their wheelhouse. So we're very happy to have them today to speak on this incredibly pertinent and central topic of homochirality concerning the origin of life. And to give the introduction is Professor Donna Blackmond, who I think needs no introduction and is a world expert on this topic. So Professor Blackmond, please take it away.
Thank you very much. So I'll share my screen. Okay, and my talk. Great, thanks. Can everybody see that? Yeah, I'm just gonna give a brief few minutes about our work in general, which we've been doing really for the past 20 or plus years on this area of trying to understand the origin of biological homochirality. And it's a topic that most of you know is fascinating, both to scientists and lay people alike. Thinking about mirror images and Lewis Carroll talking about when Alice was curious about going through the looking glass, whether her cat Kitty would be able to drink the milk on the other side. And of course, we know that if you made looking glass lactose with all the wrong hand of the chiral centers in that sugar, it would not go well for Kitty's digestive system. But it's a good topic. We use it a lot when we talk to students at science festivals and trying to get people interested in science in general and some of these bigger questions. And why is this important? This idea of biological homochirality is important because we know that the molecules that make up life are single-handed. And it doesn't matter whether the right and left are just designations that we do, like in physics, the right-hand rule, et cetera. But we designate the hand of the amino acids that make up proteins as left-handed, and the sugars that make up RNA and DNA are right-handed. And your biology doesn't make mistakes. But we know that if we actually try to do this in a lab, if I go in the lab without some way to direct these reactions, I would make a 50% mixture of each. So that's the question that we're going to hear in the two talks today, is trying to understand how did we get where we are today from where we were back then. And it's a question that actually has several parts. First, you have to break symmetry. You have to find some way of making more of one than the other. And that's a trivial question. If you think about every odd number of molecules you make, you've broken symmetry, but you have to, that goes back into the noise. So you have to find a way to consistently get symmetry to break and stay broken, but that's still not enough. You have to be able to amplify it, because biology isn't 49-51, it's, 100 to 1. So that's what we've been trying to understand. And people talk about how this could have happened. There's a lot of arguments in this field. If you go to these meetings, they're sometimes fascinating because it's almost, we're talking almost about philosophy rather than science. People have ideas about, well, maybe this actually came from outer space. You could have, we could have been seeded from outer space, which in some ways begs the question of how it happened out there. But that's one of the things that people debate. And they also talk about, was this a random choice? Or were we preordained to be left-handed amino acids and right-handed sugars? And so these are unanswerable questions unless someone here develops a time machine, but they make for a lot of very, I would say, interesting discussions at Origin of Life meetings. And so some of the idea about seeding, we know that meteorites that have been, we may have talked about this in this series, and I think we've got online one of the authors of this paper talking about the non-racemic amino acids that have been found in meteorites. And typically, like for instance, alpha valine has been found up to about 18% EE, I think. I might need to be corrected on that with latest figures, but it's not a proteinogenic amino acid. It lacks the acidic hydrogen to the proteinogenic amino acids have the alpha hydrogen. But this might be considered sort of a fossil because these molecules that are found here wouldn't racemize as easily. So they may be giving us an indication that, we receded left-handed from space. So a few years ago, and I'm not sure, we may have somebody on here from this group too, but I was just gave a seminar at Caltech 2 weeks ago, where some of the people who did this work, Jeff Blake's group, found a chiral molecule in interstellar space the first time. And I don't think there's been another one found since then. Now, they couldn't tell whether it was racemic or not because it's very difficult. You're doing this from spectroscopy, but they found propylene oxide in interstellar space. And so it got a lot of press. It was as again, I said that, this is fascinating to people that don't actually work in science or work in this field of science. And so you Google it on that day and you got, science detected this life-forming molecule in interstellar space. So propylene oxide is a compound in like green Coke bottles. So if our green Martians are made out of propylene oxide, then maybe we can call it life. But anyway, I'm not sure that how this correlates with life, but it's a fantastically interesting discovery. And then there's this idea, again, we use the handedness. We talk about non-superimposable left and right hands, life's first handshake. But sometimes I think we use that as an analogy or as a metaphor, but sometimes gets taken a little far because on the day that this came out, if you Googled this, if you went to Facebook, the trending and Facebook, it was coming up forth as scientists discover compounds resembling human hands outside the solar system. So you have this image of like, Edvard Munch, so I think that sometimes using that analogy of handedness is a little bit too, people are taking it a little bit too far. But so we are studying this chirality issue in the context of origin of life in general. And people in origin of life, I think in the past, I think this is getting a lot better. And it's because of symposia like this, where we are talking to each other from other fields. But people tended to work in silos, where they think about the monomers, amino acids and sugars, or they think about how do we make polymers, or what do we do with these polymers. They think about compartmentalization, because that's a big thing. You need to have cells. You need to be able to concentrate components and systems chemistry, how complicated things get. And the one that sort of was left out of most of those areas is chirality. So we kind of call it the elephant in the room. People can come up with lots of interesting ideas about how you can make these molecules, but we still need to make them single-handed with one hand. We can use two hands to make them, but we need to make them as one hand. So John Sutherland, you heard about it from one of his group recently, but he came up with this, which is really now, I think, pretty, I want to say universally, but we still have a lot of arguments in this field, but it's a really compelling network for plausible pathways for all the major classes. Amino acids, nucleotides, and lipids can all be made from these viable small molecule high energy precursors that we might find in outer space. So it was his first work. In 2009, he and Matt Powner published a paper where they took glyceraldehyde and they went from glyceraldehyde to RNA precursors. And this was a big breakthrough for the RNA world hypothesis because people have been trying to put ribose and a nucleobase together, and that was prebiotically really, really difficult. But bypassing ribose, they were able to get there, at least for the pyrimidines. And if you could find a way to get glyceraldehyde as enantiopure, you're home free here. But if you look at the other classes, we still have a problem because if you take glyceraldehyde and you go to the amino acids at the top right, you lose stereochemistry. And for other molecules, there was no stereochemical control. So we still need to find how we can impart selectivity in these other cases. And really what we'd like to do is to find more general solutions. We've been working for more than 20 years. We found little bits and pieces like, oh, for this amino acid, we can do this, and for this sugar, we can do that. But it's not as satisfying as trying to find a general. And I think that the two talks you're going to hear today are going to try to speak to finding more general solutions for how this problem could have started to enantio-enrich prebiotic feedstocks. So, we published a lot of work in a lot of areas, and I'm just not going to talk about it, but I'm just going to highlight the two papers in red, or what you're going to hear about from our two people today. Oops. Yeah. Yeah. Jinhan was first author on this PNAS paper, which actually is her second first author paper as a summer intern here before she started her PhD. She was first author on a paper also dealing with chirality. You'll hear a little bit about that one. And then Neal is going to talk about the paper we just published in Nature, where he pretty much single-handedly developed the kinetic model that he's going to tell you about today for prebiotic ligation reactions based on Matt Pauner's seminal studies in Science and Nature in the last couple of years, where they didn't address directly the issue of chirality. But I think this is where we're starting to come together in this field, is where we can pick up on each other's work, and when somebody comes up with a really interesting prebiotic reaction, if they haven't addressed the chirality issue, then we can look at it and sort of try to take that on. So that's pretty much all I'm going to say, and I'll turn it over now to Jinhan and Neal to talk about how we're trying to bring the concept of chirality into prebiotic chemistry. So thank you. I will stop sharing, and Jinhan can start.
I just want to make a quick note to remind everyone that we will hold all questions for both speakers until the end, and we'll have a combined Q&A session. Thank you.
Okay, let me share my slides. The students ask me who can share slides.
James, are you there? Let's see.
I don't think it allows me to do that.
It's not allowed. Let's see, because I think you are a co-host. Let me see. And no one else is sharing slides right now. Me just change something. Are you able to do it now?
Let me try again. Okay, yeah, it's okay now. Can you see my slide?
Yes.
Okay. Good morning, everyone, and thank you, David, for your invitation, and thank you, Donna, for the introduction. It's my honor to be here today. Today, I'm going to share with you our work, prebiotic access to enantium enriched amino acids with peptide-mediated transamination reaction. As you just heard from Donna, the origin of biomolecular homochirality is an unsolved problem in the study of the origin of life. In the early Earth environment is most likely to have no bias towards one hand between the two. How can we get enantium-enriched materials from racemic feedstocks and eventually achieve biomolecular homochirality in the preparation of life? As you just heard, the biomolecular homochirality concerns two families of biomolecules, sugars and amino acids. When we first started this journey, we asked this question: can we use L peptides to catalyze the formation of these sugars? Because since we have two families of molecules, how does that happen? That's probably because it might be because they have some interplay of chirality information. And we use peptides here as a catalyst because peptides can serve as a very primitive form of enzymes. This is a previous work from our group. It's done by former postdocs Alex and co-authors. In this reaction, they discovered that L peptides can catalyze the self-condensation of glycoaldehyde to make a 4-carbon sugar, and is enantium enriched in the biological enantiomer, the D enantiomer. When I first started work on this, our question is: can we extend this reaction to form 5-carbon sugars using glycoaldehyde and glyceraldehyde? Can we achieve enantiomer-enriched D-ribose using the same series of L peptides? The answer is no, because the L peptides Instead of catalyzing the reaction, they react with glyceraldehyde. So we first look into this interaction. It turns out that L-peptides intend to enrich the glyceraldehyde. This reaction proceeds as a kinetic resolution, as you can see here. L-peptides selectively react with L-glyceraldehyde, the non-biological enantiomer, faster, and it gives this Amadori byproduct. The D-glyceraldehyde is left over and then enriched. By monitoring the leftover of glyceraldehyde, we can see that E equals D increases as the reaction reaches higher conversion. Here is an example, and this example shows the interplay of chirality between the peptides and sugars. Another interesting point is that here we have biological enantiomer enhances another biological enantiomer. This is a good news for achieving biomolecular homochirality. Based on this example, we learned that L-peptides intend to enrich these sugars. And then we ask, what else can we do with these L-peptides? Can we use the peptides to enrich the other category of biomolecules, the amino acids? That's how we start to look into the transamination reaction. We know that transamination is an important enzymatic mechanism. It's basically an interconversion between the amino acid and alpha ketone acid to generate a new alpha ketone acid and a new amino acid. This process is mediated by the transaminase and its cofactor PLP. Transamination produces most of our non-essential amino acids. Here in this reaction, the same R1 amino acid can produce many different R2 amino acids while retaining the R1 reality. So we first have the idea of using a prebiotic peptide catalyst for transamination that would allow one amino acid to transfer its chirality to others. This idea is especially attractive as it will free us from the burden of solving the enantiomer enrichment of proteinogenic amino acids one by one. That will be a total of 20 cases. So we first look into the enzymatic mechanism for this reaction. Here, the cofactor of transaminase, PLP, binds the substrate amino acids to form the intermediate, and it tautorized to give this achiral intermediate. The chirality information of the amino acid here is lost. The intermediate then breaks down to release the alpha ketone acid with PLP converted to PMP. The amino acid in this step is a sacrificial reagent. As to produce amino acid, we need nitrogen, and this amino acid serves as a nitrogen source. And here we reach PMP. PMP will take the second substrate, the alpha ketone acid, to form a similar intermediate, also achiral. To restore the chiral information, we will need an external directing template. Here within the enzyme, we'll have the lysine residue to direct the proton transfer exclusively for one phase, and therefore a new amino acid with single and correct chirality is produced. So in the enzymatic mechanism, the enzyme enables the production of enantiopured amino acid. Examining this cycle, we can see that it actually consists of two half reactions coupled together. One going forward from alpha ketone acids from alpha ketone acid and predoxamine to produce amino acid, and the other going backward with a sacrificial amino acid converting PL back to PN to keep the cycle running. So to make the question simpler, we look at this half reaction and we ask, can we use small peptides to enable the production of enantiomer-enriched amino acids? So we start with this model reaction pyruvate as the alpha ketone acid and pyridoxamine, the unphosphorylated version of PMP. And these two make alanine and pyridoxal. We tested a series of peptide catalysts. What we found here is that in many cases, we do have enantioselectivity, but the proteinogenic LL dipeptides produce non-proteinogenic D amino acids. The best catalysts are the dipeptides with proline at N terminals, reaching up to 50% EE towards D enantiomer. These results are validated by using the opposite hand of the catalyst, and as expected, it gives opposite result. Another interesting thing is that by flipping the second stereocenter on the dipeptide, it basically destroys enantioselectivity, since the LD proline gives no EE at all. So we have a problem here. Although we achieved enantioselectivity, now the biological enantiomer is enhancing the non-biological enantiomer. This will not be good for biomolecular homochirality. So how should we solve this conundrum? We have a hint from the principle of microscopic reversibility. The principle of microscopic reversibility is that if one reaction goes somewhere, the back reaction will have to come back in exactly the same pathway. Here in this case, we know that L-peptides produce D-alanine faster than the L-alanine. Then in the reverse reaction, we can expect that D-alanine will go faster back to pyruvate than L-alanine. If we start with equal amount of L and D-alanine, the same L-peptide will consume D-alanine faster than L-alanine, and that would be good news for biomolecular homochirality, because here we'll have the biological enantiomer enhancing another biological enantiomer. With this hypothesis, we ran the reaction in reverse, starting from racemic alanine and pyridoxal to make pyruvate and pyridoxamine. And this time, indeed, among the leftover of the alanine, the L enantiomer is indeed enriched, and the best catalyst is still the peptides with proline at the N terminal. This result is also validated by using the opposite hand of the catalyst, DD proline valine enhanced D-alanine. Now here we get the correct result. So we started looking into the reaction mechanism of this network. At first, we prepared the equimolar of the four component alanine, pyridoxal, pyridoxamine, and pyruvate in the absence of catalyst. We just mix these four and watch them equilibrate. Upon mixing, a number of secondary species was observed. Here we call them A, B, and C. And we thought, and we chased its formation. First, we think PL and alanine formed species A and albumin. And second, PM and pyruvate formed species B and a cyclized hemiaminal. This is probably formed by the intramolecular nucleophilic attack of the ketamine. The ketamine is not observed, probably since the equilibrium draws towards the species B. And third, PL and PN condense to give species C. The two parts are probably linked by the tautomerization between aldermine and ketamine. And the highlight part is the main pathway of the transamination reaction. And then we ask, what happens once the catalyst is added to the network? To answer this question, we conduct a similar reaction. We prepared a mixture of all four again, pyridoxal, alanine, pyridoxamine, and pyruvate. 50 millimolar, and then this time we add catalyst into the mixture. And to be able to trace the forward and the backward reaction simultaneously and separately, we labeled alanine and pyruvate as different carbon, and then we monitor the reaction with carbon NMR. So first, let's take a look at the forward reaction. As we can see, species B builds up at the very beginning. That means the equilibrating from the pyridoxamine and pyruvate to species B is a fast process. And over time, we can see the alanine and A accumulate gradually. And that suggests from ketamine to the rest of the network, it's comparatively slow. We then look at the backward reaction, starting from pyridoxal and alanine. And this time we can see species A builds up at the beginning. And that suggests from pyridoxal and alanine to aldermine, it's a fast equilibrium. And we can see here that pyruvate forms very gradually over time. And that suggests that the rest of the network it's lower compared to the first equilibrium. Combining these two results, we can conclude that the rate determining step of this network is the tautomerization between aldermine and ketamine. So far, we have not talked about the catalyst yet. So what role does the catalyst play here? Our hypothesis is that since the substrate rests as the aldermine is probably the species to be captured by the catalytic cycle. As we have already know from our experimental results that we have selectivity, then the aldermine is probably captured into this catalytic cycle slower compared to that of the aldermine, and that's probably why we have enantiomer selectivity. To delineate the origin of enantiomer selectivity, we conduct computational modeling. This part of work is done by Andrea, a former postdoc in our group. Here, starting from alanine and pyridoxal, we can see the energy of aldermine is lower compared to the two substrates. This consists with our experimental observation that aldermine builds up at the very beginning. The next step is the tautomerization from aldermine to ketamine. There are two possible configurations of ketamine, Z or E, corresponding to two sets of mechanisms. The Z pathway, the black one, and the E pathway, the blue one. The enantiomer determining step is when the catalyst removes the proton from aldermine. We can see that the energy barrier of the D enantiomer, ST1ED, is lower than that of TS1EL. And that's why in the transition stage of STED, we can see the additional interaction between the two parts. Here, there's an additional hydrogen bond between the carboxylate group of the of the peptide and OH group of the albumin. Such interaction is not available in the transition states of the L enantiomer, the SD1EL. Therefore, the lower energy barrier on D pathway is due to the additional hydrogen bond interaction. And after the enantiodetermining step, the two pathways merge to the same intermediate 1E, which is a high energy species that's not observable by experiment. And the catalyst would deliver the proton back to the substrate and release the ketamine. The ketamine then breaks down to give pyruvate and pyridoxine. The last part of this work concerns metals. Metals have been a much discussed topic in prebiotic chemistry because of its connection to modern biology. As we know that many modern enzymes possess metallic cofactors for structural or functional purposes. So at some point of the prebiotic history, metals will have to come into the picture. A recent work by the Moran group reports that metals accelerate rates in transamination reaction. With this background, we test metals in our system. So we add 10% of metal to the peptide-mediated kinetic resolution. And we monitor the conversion of alanine and EE of the leftover alanine. The purple curve represents the blend control with catalyst, but without metal, and the other three are copper, zinc, and alumina, respectively. Here we can see very clearly that in the presence of metal, both reaction rate and enantioselectivity suffer. We then mix metal directly with enantio-enriched alanine to test the stability of alanine EE in the presence of metal. Here again, when there is no peptide and no metal, there's no EE erosion in alanine. The peptide itself erodes alanine EE as it brings the system towards equilibrium. And in all cases with metal, the EE decreases. So while metals alone do not induce enantiomeric imbalance, they further cause racemization of the enantio-enriched alanine. Metals can have different roles in different reactions, but here in this case, metals are detrimental to the reaction. So here is the conclusion. In this work, we discussed a dipeptide catalyzed transamination of racemic amino acids mediated by pyridoxal, and it leads to enantioenrichment of the proteinogenic L amino acids via kinetic resolution. And kinetic resolution of racemic starting materials can be a great tool to achieve enantioenrichment in building blocks of life, as we have known that early Earth probably starts with racemic feedstocks. Based on this primitive form of transamination reaction, based on this primitive form of transamination reaction, we can achieve some enantio-enriched building blocks, as all these primitive pathways accumulate enantio-enriched materials, making it possible for prebiotic chemistry to evolve to a more sophisticated stage. More potent catalysts can be developed to really enable the chiral transfer among the amino acids. That is, to close the cycle of the transamination reaction. And that's probably what becomes today's today's enzymatic the enzymatic mechanism. And this example of kinetic resolution joins the other 4 previous work from our group, and they are all kinetic resolutions. The last one is the one you will hear from Neal. Now, all 5 examples of kinetic resolution suggest that kinetic resolution of racemic starting material can be a general approach to achieve enantiomer enrichment towards the emergence of homochirality in the building blocks of the molecules of life. And here's my work. I would like to thank my advisor Donna and my coworkers. I would also like to thank Scripps Research and Simons Foundation for their funding. I'm happy to take questions in the Q&A section.
Jinhan, it was incredibly interesting. Neal, whenever you're ready, take it away.
Okay.
Let me stop sharing.
I guess you can see my slides, right?
Yes.
Okay, cool. Good morning, everyone. I would like to thank David and Donna for the introduction and Jinhan for a good transition to my talk. And today I'm going to talk about symmetry breaking and chiral amplification in prebiotic peptide ligation. So first, I'd like to show you this beautiful artwork done by David Goodsell, a associate professor from Scripps Research. And here you see is a water painting of E. coli. And this in this cell, you can see a lot of large molecules, and these are made of biopolymers. And we all know that living organisms can produce all kinds of biopolymers, such as proteins and nucleic acid. And these molecules are usually single-handed. And to make these large biopolymers, taking protein, for example, it should start from simpler molecules like amino acid. And then you assemble the simple building blocks together to first make some peptides and then further extend the chain to make the protein. And we know that all proteins in cells are composed of left-handed amino acids. In modern biology, this is easy because we find all the building blocks left-handed. You just need to assemble them together, then you get left-handed polymer. And if you don't get any racemization, then you don't need to worry about anything at all. But in prebiotic chemistry, it's likely that we start from racemic or near racemic building blocks. So the question is, how did we get here from there, from a racemic or near racemic prebiotic soup to now a homochiral modern biology world composed of all kinds of biopolymers? In order to do that, there are a couple of steps that we believe nature has to overcome. First, as Donna mentioned, we need to have symmetry breaking of molecules, of chiral molecules. And to do that, there are many ways people have proposed, like the previous talk from Furkan, a graduate student from Harvard University. They mentioned that magnetic surface can break symmetry of RAO. And then there is also circularly polarized light, people believe can break symmetry. And then when this is done, you start from a balanced 50-50 mixture, and then you get 49 to 51 imbalance. That's not enough. We need to amplify this imbalanced enantiomeric excess to a higher value. And to do that, there could be chemical process or physical processes to enrich higher enantiomeric excess in the end. And starting from there, when we have one or a couple of enantiomer-enriched molecules, we need to propagate this chirality information from one to all the rest of chiral building blocks, and that we call chirality transfer. And in modern biology or modern chemistry, we can do that easily with asymmetric catalysis or dynamic kinetic resolution, etc. And then in order to find chiral amplification process, our lab, as Donna mentioned, have been working on this for more than 20 years on various kind of models, starting from pure chemical process to pure physical process or combinations between them. However, there are two big challenges still in these models. First, these models don't always end up with perfect enantiomer purity, and it's usually tailored compound by compound. So this leads us to two other questions. How can we break symmetry of all kinds of chiral building blocks to the same direction at the same time? Because otherwise you won't be able to make a homochiral biopolymer in the end. And the second question is, had nature waited until every building block got enantiomer enriched to start forming functional polymers? So these are the questions we had when I started my new project. And then in order to do that, we got to find the prebiotic chemistry, the ligation chemistry that can connect monomeric building blocks together and to form polymers in prebiotic conditions. And then we also need to be able to track the stereochemistry from the monomers to the products. And if we could have that kind of reaction, then we could probably start exploring the stereochemistry in the process of forming higher order polymers. This reaction, published by the Towner group since 2020, really caught our attention. This reaction uses an amino nitrile and an amino acid to make peptides, simply catalyzed by a thiol. So with the thiol in this reaction, you can string amino acids building blocks together to make higher order chains. And the stereochemistry from the amino acid building blocks can be retained in the product. So we wondered, in this reaction, both substrates can be chiral and the catalyst can also be chiral. What kind of diastereoselectivity could this reaction have? Is the chirality in catalyst important for this reaction? Then we carried out a series of diastereoselectivity studies using either racemic 1 enantiomer 2 or vice versa. In studying this, we can see whether you use enantiomer 1 racemic 2 or racemic 1 enantiomer 2, we get similar results in the diastereoselectivity. And we also observed that when you use a comparing to the chiral catalyst 4, when you use an achiral catalyst 5, we got very similar results, which shows that the enantiomer purity or chirality standard in the catalyst doesn't really matter for this kind of ligation. It's purely substrate controlled for the diastereoselectivity. Then we further expanded this reaction to various kind of chiral amino acids and even peptides or amino amides. What we found that is there is all kinds of different diastereoselectivities with different kinds of amino acids. And then we want to know which one is the major and which one is the minor, because you have diastereoselectivity, but it doesn't necessarily mean if that's the homochiral is the major. Then we synthesized enantiomer product to compare, and we found out all of these ligations favor a heterochiral ligation. The heterochiral product, DL or LD product, is the major ones in the peptide product. And this shows us that this reaction is purely substrate controlled. It's probably a good thing because prebiotic chemistry doesn't guarantee enantiomer high EE catalyst. So using achiral catalyst or racemic catalyst, we can achieve diastereous activity, which is a good thing. And then this reaction all favors heterochiral ligation. Is that a bad thing? Maybe not. So in studying this reaction later, we found out there is a couple of amplification mechanisms going on in this type of ligation. We first started with two simple ligation reactions with non-enantiopure, non-racemic starting substrate. The first reaction, we took leucine amidonitrile with 20% EE, and then we added 20% EE alanine into the reaction. And the second reaction, we switched the alanine to 20% EE valine. What we found out at very low conversions of this reaction, there is already 40% EE in homochiral product 3. So this kind of caught our attention. Why is this happening? And by analyzing this reaction, we know that when you have all 4 enantiomers in this reaction, you can do 2 homochiral ligations to make the LL or DD product. And then you can also have reactions to make LD or DL product. And with the analysis of this simple second order kinetics, and at the beginning of this reaction, we know to measure EE or to calculate EE of homochiral product 3, we can use this equation to represent how EE of homochiral product 3 is measured. And then this reaction can be replaced by replacing with the K times the concentration of the starting materials, and replacing and you can cancel out the K homo here, and then replacing the initial concentration of L or D with EE1 and EE2 and the initial concentration of total concentration of 1 and 2. You can get this equation, which is composed of starting initial EE from 1 and from 2. And we call this equation equation A. And with that, we can probably predict all kinds of same EE ligation reactions with different starting EE. And we use this equation, we got this gray curve as the plot of homochiral product EE versus the starting material EE. And that gives us an intrinsic amplification. However, when we carried out our reactions and we measured the EE of homochiral product, which is even higher than this, you may wonder, is this just experimental error? We also wondered that. So we carried out the experiment with 50% EE of both substrates and monitored the EE change of homochiral product over time. As you can see from this figure here, when you increase the conversion of the reaction, you get higher EE values of homochiral product. That means this is definitely not an experimental error. That is something going on in this reaction. And why is that happening? We took this reaction and we further monitored the substrate EE over as the reaction proceeds. As you can see, both substrates EE starting from 0.5 gets amplified as the reaction proceeds to full conversion. And the dots shows our experimental results. And we also used a software called Copacity to model this reaction. We got this blue curve, which match with our experimental results. And then you probably also wonder, this is happening with a heterochiral preference in the ligation. What if you change this to no preference in the ligation, or you make the reaction homochiral preferred? With our software, we also predict the other two cases. As you can see from here, the gray line shows that when there's no preference in this ligation, the EE of products or substrates doesn't change from time zero to full conversion. However, with homochiral preference here, you can observe a chiral information depletion process or EE depletion over time. So that leads us to find out this 3D structure. This 3D figure shows that when you feed this reaction with various combinations of EE1 or EE2, what's going to happen for a homochiral product there? As you can see here, without any preference in the ligation, there is a surface with 1-to-1 K hetero to K homo generates the homochiral product EE on this surface in the middle. And with the heterochiral preference, the homochiral product EE sitting on top of this surface. And with homochiral preference in the ligation, it's all below this 1-to-1 surface. The higher the diastereo's activity, the more amplification or depletion you observe in this reaction. And taking a slice from the middle, you can see this more clearly. When you have same EE ligation, same starting material EE for both substrates, you get all kinds of implication. Depends on how much heterochiral preference you get. Although you still get a little bit ligation, you still get a little bit chiral amplification with the homochiral preference. But in the other case, when you take a slice from the side, where you start from one racemic substrate and the other non-racemic, non-enantiomer, then you get this kind of curve. You can only observe chiral amplification when there is heterochiral preference in the reaction. And when there is homochiral preference in the reaction, there is definitely a depletion of EE in the homochiral product. And with this, we now know that heterochiral preference is great because it leads to chiral amplification through two mechanisms. First, an intrinsic multiplicative amplification, and then a temporal amplification as the reaction proceeds. And then we further considered in prebiotic chemistry, it's probably not just two components in your prebiotic soup. From a systems chemistry perspective, you would wonder, what if when you have more substrates in your reaction, for example, we designed this reaction with one aminonitrile and two amino acids, what is going to happen in this reaction with the heterochiral preference ligation? And when you have three components as substrates, the elementary steps in this reaction is a bit more complicated. Then now you have 4 homochiral ligations and 4 heterochiral ligations going on. And then with that, we kind of design a reaction that we want to start with. In prebiotic chemistry, as we mentioned earlier, it's likely that you have 1 or 2 components that's already symmetry broken, and then the rest is racemic. In order to mimic that kind of condition, we wanted to start with 0.5 EE for one of the substrate, like 2, and then we treated the other 2 substrates as racemic in the beginning. And what we wanted to observe is the experimental results of EE of all components in this reaction. And we'll also want to do the modeling to compare with our experimental results. And with this reaction, it's not that hard to predict that when 1 and 2 react to start generate product, the EE of 2 gets amplified over time. And this can be transferred to the EE change in 1 as well, just like our 2 component reactions, right? And then what is interesting here is that when you look at the other amino acid substrate in this reaction, the EE of this component is also getting amplified over time, although it doesn't interact with the non-racemic component 2 directly, right? And then not hard to see here is that we found chiral amplification from this non-enantiomer component. And this component can cause symmetry breaking in its coupling partner. And further, this chiral information is transferred to the other amino acid component. And with this, it's not hard to predict that the product, homochiral product EE, also gets amplified because substrate EE gets amplified over time. So the product EE also gets amplified over time. And our prediction from these blue curves match very well with our experimental result of the yellow dots. And from there, we know that only one non-racemic component is actually required to achieve symmetry breaking, chiral amplification, and the chirality transfer in this kind of ligation reactions. And the next thing I want to bring up here is to understand this heterochiral ligation better by looking at a simplified model. We want to go back to this two-component reaction again by looking at three different cases, like no preference in the ligation, or high homochiral preference, or high heterochiral preference in the ligation. These these preferences are far from our actual reaction conditions because we treated it as like a super high homochiral preference or super high heterochiral preference. So it's not ideal, but it will help us to understand how everything processed during the reaction. So let's start with racemic 1 with EE0 and 0.33 EE for the other substrate. And what is gonna happen when you have no ligation preference in this reaction is that you would take 2/3 of the racemic one to react with all the L's from the other component. And the leftover racemic one will react with all the D's from the leftover 2. And this will result in 0.33 homochiral product. And there's also heterochiral product generated, which also has 0.33 EE, but we don't care about this part very much because it doesn't compose homochiral chain. And then when you consider this reaction extremely homochiral preferred, then you would generate as many homochiral peptides as you can. And then in the end, you only have one D and L left from each of the substrates, and you force them to make one heterochiral product. And in this case, it will result in EE depletion in the homochiral product from 0.33 to 0.2. And similarly, when you treat the reaction with extremely heterochiral preference, what you get is you get as many heterochiral products as you can, and then the leftover is only 1L from 1 and 1L from 2, and then you force them to form one homochiral product, and then you make this homochiral series enantiomer as a chiral amplification process. And if you look at all this process together, you can see when there's no ligation preference, then there's no amplification in the homochiral product when you ligate with racemic 1 and non-racemic 2. And then when there's extreme homochiral preference, then you get EE depletion of homochiral product by generating a large amount of homochiral product. And then when you have strong heterochiral preference in the ligation, you can amplify your homochiral product EE by generating a large amount of heterochiral product. You may wonder if this is the real case, then we generate so much heterochiral product in this system. Is that a bad thing again? And then in studying this reaction, we found out nature probably has a solution to fix this kind of problems. So comparing here, we have two reactions. The first one, we start from racemic 1 plus enantiomer 2 to simply observe the kinetic resolution of 1 by 2. And here we generate a lot of DL heterochiral product, more than homochiral product. Over time, both concentrations increases. However, when you start with racemic 1 and racemic 2, where you have all components of L and D from both substrates, then you can still form heterochiral product and homochiral product. And here you have both LLDD and DL and LD. And in this case, in the first 2 days, it has a similar trend. Both concentrations for homochiral and heterochiral products increases. And then on day 3, the heterochiral product concentration drops immediately. And this leads to like an inversion of homochiral to heterochiral product ratio. And this shows that the precipitation of this process removed most of the heterochiral product from the solution. And with the X-ray characterization, we found out that actually this crystallization removes one DL and one LD simultaneously from the solution to make this one-to-one packed insoluble rockish crystals. And when you generate these rocks in the solution, it's really become really hard to redissolve them back in solution because they form very strong hydrogen bonding between each other, and they don't want to get redissolved anymore in water. And then taking this solution, we decant the supernatant or the solution from this vial and the adjusted pH and temperature. And a second precipitation event happened, which generates LL and DD one-to-one packed crystals. As you can see from the vial, these crystals look different than the heterochiral ones. It's more like a needles here. And with that, we think we can probably combine all this chemical amplification process and the physical process together to amplify EE starting from low EE substrates. So we designed a reaction starting with 20% EE of 1 and 20% EE of 2 and let them ligate with a heterochiral preference. And the multiplicative and the temporal amplification mechanism can lead the EE from a low value to a higher value. Here we can get 55% of homochiral product EE. And with the first precipitation event happens in the reaction condition, we can remove most of the heterochiral product from the system. And what's remaining in the solution is mostly homochiral product with 55% EE still. And then the second precipitation event could happen if we adjust temperature to 25 degrees of Celsius and the pH to PD to 8.5. Then we get the second precipitation event, removing one-to-one DL and LD from solution to further amplify the EE of our homochiral product through a physical process. And overall, studying from just 20% EE of each substrate, we can end up with 81% EE in the homochiral product. And that's what we observed with our experiments. And in order to summarize all this work, we know that in prebiotic conditions, we probably have all kinds of chiral amino acid feeding stocks together in one prebiotic soup. And in this soup, they ligate, they react, and forms a kinetic resolution network in the process of generating biopolymer reaction. And then through chemical process, we know there are two amplification mechanisms can lead to symmetry breaking and chiral amplification and chirality transfer in monomers and polymers. And with physical process, we can further purify and amplify the homochiral product to an even higher enantiomer enriched status. And from there, we could probably start having homochiral functional polymers and be ready to for the emergence of life. And with that, I would like to thank a lot of people that helped me during the work. First, of course, Donna, and then I would like to thank Jinhan helping developing some critical EE measurements methods, and our AES automatic synthesis facility lab from Scripps Research to measure some critical EE values. And I would also like to thank UCSD XRD lab for helping us getting the crystal structures. And one of my friends from University of Missouri helped us producing the beautiful 3D figure in this paper. And I would also like to thank Simon's Foundation for supporting our research. And that's all I want to say. And thank you for your attention, and thank you for having me here to give this talk to you. I would like to take any questions if the audience would have. Thank you.
Thank you very much, Neal. That was a very interesting talk as well, and it's our pleasure to host you for this seminar. So for the Q&A session, I just want to give some direction to everyone, since I think there will probably be a lot of questions. You can raise your hand, and we will call your name, and you can ask your question out loud, and you can unmute yourself and speak it, or you can put your question in the chat and we will read it aloud, and I will do my best to choose everyone based on the order in which they introduce their question or raise their hand. Well, maybe we'll just give it 1 second. Well, I was interested in the possible interplay between the two mechanisms that you have demonstrated in these two bodies of work. Now, I know that, in the Blackmond lab, there have been quite a lot of different mechanisms proposed for EE enhancement, possibly acting in a way that enhance each other. Do you think there is any possibility for interplay between these two different mechanisms of EE enhancement?
Me? Yeah, I think definitely all kinds of mechanisms can play together, right? Because we can only imagine. We don't know what actually happened in prebiotics too. So all kinds of mechanisms, if the condition allows, they could help each other and amplify each other, and therefore, in the end, it could be, just all together processing, forming the homochiral polymer or homochiral molecules.
Donna, did you want to comment?
Yeah, I think one key point in these two talks, in any talk about homochirality, where you have to have some symmetry broken component to get started, right? And so inevitably the question is, yeah, but where did that come from? And so you see Jinhan started out with these dipeptide catalysts, which gave her amplification first for glyceraldehyde in her first paper, and then now in the transamination. But then you say, well, where did she get that? Now we turn to Neal and he can give us a dipeptide catalyst. So I think these things are almost like, I don't know, leapfrog or chicken and egg, we don't know where the first seed of asymmetry came from, but once we can make once we can make dipeptides with some enantiomeric excess, then we can do reactions with them like Jinhan showed. So I think that's where the interplay comes in, is that we may never solve where the first excess came from, but we can then use that, to get us other places. And so it helps to solve that question that comes up. Naysayers often, when you're giving a talk, say, yeah, but how did you get that one that you're using? You started with 20% EE this, where did it come from? So I think that the interplay in these two talks, helps to show that.
Yeah, agreed. And I mean, you mentioned in your introduction, and I think I've heard you say this before, and I totally agree. It's like if you're thinking about the dynamic process of an asymmetric reaction, every odd reaction that occurs necessarily breaks symmetry. So it makes me think, if we could go back and play the tapes again, not just once, but 100 times, would we get 50% of the time it would be the opposite of what we have now? Or yeah.
Yeah, I mean, I think the key there is how you can get above noise. I think if you did it millions of times, you'd break symmetry 59, 41, 41, 49, 51, 59, 41, 51, 49. But at some point it had to jump out, and we treated that, for instance, in the SOI reaction, cases like that where it escapes, or in the Viedma ripening, where you can escape that going back into the noise. I mean, it's trivial. Every other reaction, when you have like 6.10 to the 23 molecules per mole, one of them, one uneven isn't going to help you. But I think, I mean, so your question really relates to one of the sort of big philosophical arguments, which is, could we have gone the other way? And some people swear they think we had to be left-handed amino acids, and that's it. Some people swear it was a random choice. I think the only thing that our work, not just these two, but the other work shows, is that once you pick L amino acids, you're picking D sugars, or vice versa. You're not going to pick L amino acids. Our form of life wasn't going to pick L amino acids and L sugars. I mean, that's, I think, pretty clear from the results. But how that happened, whether it's random or preordained, it's beyond my pay grade.
Very interesting finding, though, that the two are inherently coupled. So.
Yeah, we're not the first ones to say that, or that there's a lot of sort of theoretical work kind of showing that. And I think I think it makes sense.
I think we have a question from Eduardo. If you want to go ahead and unmute yourself and ask your question.
Okay, can you hear me now?
Yep.
Yes.
My question for Jin, there's two for you and two for Neal.
Yeah.
First, what was the pH of the sorry, a little nervous. The pH of the medium used in the first experiment.
Yeah.
And then to follow that up, at the end of the experiment, did you continue to have dipeptides? Did you ever have any more peptides, longer peptides at the end of the experiment?
Okay, so I think your first question is about what's the pH of the experiment and why is that?
Right. Yeah.
So the pH is mildly alkaline, pH 8.5. Why is that? It's because we test a series of mild pH that we think is prebiotically possible, from mild acidic 6 to a more basic like 9. And 8.5 gives us best result. If we compare this pH to the pKa's of the pyridoxamine or pyridoxal, it actually corresponds to the pKa of the nitrogen on the aromatic ring. So we think that might help in the pH is the best one in our screening reaction because it helps all this proton transfer process. And your second question is did we try longer peptides?
No, if at the end of if I know you started with dipeptides.
Yeah.
At the end of the experiment, were there any? Did you check the resulting products to see if those dipeptides became longer peptides?
No. So the process is catalytic. So the peptide acts as a catalyst. it's not the reagent of the reaction. After reaction is done, it's still the peptide. We do produce, we do have amino acids around in the system, but under this condition, these amino acids is not going to ligate with peptide to give a longer product.
Okay. And now my question for Neal. Is
Yeah,
there
go ahead.
any is there any knowledge on, is there any sequence of amino acids that can produce other amino acids that are not left-handed? If you understand what I mean. Is there a sequence that life produces left-handed amino acids? Have we ever seen that? Is there a possibility of life producing left-handed amino acids by just a change of the sequence? Because I'm wondering, is life only using one-handedness because that was the sequence that appeared first during evolution, or during the initial production of enzymes to produce amino acids? You understand what I'm trying to get at?
Yeah. I think you're asking why life only chooses left-handed to make a left-handed protein world, but not a right-handed, right?
Do we ever find a sequence of amino acids that can produce left-handed amino acids? I mean, right-handed amino acids. I mean, do the opposite.
Can you clarify if you're talking about with modern enzymes?
Correct. Did we find any modern enzymes that can carry out?
I mean, I think people can look at non-natural amino acids and try to look at that question. But with natural amino acids and natural enzymes, we sometimes get post-translational modifications where
Correct.
an L turns into a D amino acid in a peptide or a protein. But your body almost never produces as the when you're making an amino acid, like for instance by the transamination pathway, I don't know of any case ever where it's made a D amino acid instead of an L.
Right. Well, my question is it even possible? Maybe the reaction centers that are created by proteins can only orient it in a way that they can only produce.
That's the key. Yep.
Okay.
That's the key. I think, we're trying to get to, I mean, the 3D structure of enzymes really dictates, because in that transamination cycle, you lose the chirality, it's gone. And then you pick it back up again at the end of the cycle because of the pocket of the enzyme.
Right.
Chirality lost and found, and it only finds it in that one direction because it's what it's trained, learned to do over, eons.
Well,
Yeah.
following up, so
Yeah,
that's what I'm wondering.
I
Could we produce an artificial enzyme that could do that? That's my question.
I think it's not our work at all. Somebody else would have to
Right.
introduce that.
Yeah.
But I don't see any reason why not. If you could come up with a tertiary structure of a sequence of amino acids, maybe they would be left and right together that could give you something. But it's, and some people might be looking at, you could use directed evolution. I mean, George Church is trying to make a whole world of the other side, and maybe in between those two worlds there'd be some way to make, but
Okay.
I don't know of any work showing that.
Okay. Thank you.
Small thing to this. I think the question of what can biology do today and what happened prebiotically are two different questions. But there are examples of opposite-handedness amino acids, like in some in bacterial cell walls. There's amino acids D sugar. So there's D-alanine in some bacterial cell walls, but that is produced by an evolved enzyme, which is not the same as a prebiotic process. So is it possible for modern enzymes to produce the less prevalent handedness? Yes, but that's not, it's not a counterexample to what we're talking about today. I believe Sure Shock, you can go ahead and ask your question now.
Hello everyone, and I am Shishak from India, and I really get a question in the middle of the presentation that Neil shows. Uh, in the presentation, it means he told that there is a problem which arises in the experimental conclusion, which has the solution by nature. So I want to say that what can be the is it hypothetically something that means how the solution is getting cured by means how the solution is coming in by the nature.
So,
You
sorry, I'm
have to
not,
put cut off, yeah.
I'm not sure I understood the question. What was,
Sorry, I'm not clear on it either. Could you rephrase, perhaps?
I want to say that in the middle of the presentation, there a problem arises. Actually, that time I get a little confused that what was the actual problem, and that means the experiment has failed.
The actual problem experiment fixed. Are you talking about the physical processes? So
Yes.
yeah, so in the ligation, right, we know that with the heterochiral preference in the ligation, it's good for EE amplification, but it also generates a lot of heterochiral ligation product, which is not what we want in modern biology or for life, right, as we know. So that might be a problem, because when you have a solution full of unwanted molecules, it's probably gonna be a problem to, like stop life forming or stop some kind of reactions or like that. So what nature fixed was that there is a precipitation event going on to remove that all unwanted molecules from solution to make a solid. Did that answer your question?
Yes, that was my question.
Okay.
Thank you.
You have another question, Eduardo? Oh, you know what? There's a question in the chat that I think we should address. A question regarding homochirality as a biosignature. Could you be sure that it is a biosignature? Now, I'm not sure if this is the exact forte of our speakers, but I'll read it. Can you imagine any mechanism or phenomenon where non-biological reactions produce homochirality?
we just showed you two of them.
Yeah, yes.
We have to get from there to here, and it's going to be abiotic on the way, to getting to biotic. So but the question of biosignature is an interesting one, because you probably in this series have lots of astronomers that use this, and they talk about biosignatures into outer space, for instance. We've never seen chiral molecule. The first chiral molecule, we didn't even know whether it's racemic or not, was found in interstellar space 8 years ago now. But so to talk about biosignatures, that's a word that I think is overused. We don't know what a biosignature is. All we can say is there's a correlation between, our form of life and the fact that, left-handed amino acids, right-handed sugars. And it's not clear. I mean, if I saw, and so people have asked this, we used to have these kind of discussions in the Simon's meetings, what would you consider if you found, 20% EE of some compound, chiral compound on Mars, would you say life was there? And I don't think so. I think we'd have to actually see some kind of polymer that was homochiral before we could actually think about there being any bio somewhere else. Pre-bio, maybe.
We might have time for one more question, but just to respect the speaker's time constraints.
So there's one comment in the chat that I think we passed over, and there's several comments with talking about papers or books that we can read. But somebody asked, I have a doubt, how can all these things help us? And I'm not sure what that means. Help us solve world hunger? Maybe not. So I mean, this is pretty much so most of what we do comes from fundamental questions. So that was really the key to the Simons Foundation. You know, Jim Simons, who recently passed away at age 86, an amazing man, asked the question, you know, what are some of the fundamental unanswered questions in science? And he's been for more than 10 years funding research to try to answer these. And how does that help us? Well, some of the physical methods that we've used have actually now been used in making blockbuster pharmaceuticals, the precipitation methods that we discovered. So often very fundamental research can lead to practical things that you never thought were on the cards when you started out. Just like we started out thinking we were looking for homochiral polymers because that's what we needed to make. And all of a sudden we got the wrong answer. And it turned out we learned more from the wrong answer about how we may have got here than we would have if we had the answer that we thought a priori was right. So I think it's just a pitch for fundamental science that, you know, there's lots of, you know, if you're asking fundamental questions and you're doing fundamental experimental computational research, I think that, you know, and especially interdisciplinary, you can learn, you can ferment a lot of new ideas that will lead us to lots of things that we can't predict right now.
Referring to attrition enhanced deracemization?
Yeah.
Yeah, that.
I mean, we looked at the
Yeah,
blockbuster drug Plavix. You know, it's you know, it's anyway. There's a number of cases like that you know, Jason Hein has been showing amazing crystallization work. He did that first work on Plavix in my lab.
Thank you so much for your time, Professor Blackmond, Neal, Jinhan. An amazing session, amazing presentations. Thank you, everybody, for attending this third session of the PCE3 seminar series. And please be on the lookout for our email and Twitter announcements about the next session. So thank you all very much, and we'll call it there.
Thank you.
Thank you.
Thank you.
Okay.
Bye.